Iron in the human body is regulated by the enterocyte, as the body does not have a way to eliminate iron. When iron is needed, ferric iron is reduced to ferrous iron by ferric reductase enzyme duodenyl cytochrome B, which is located on the apical membrane of the enterocyte. Ferrous iron is then able to be imported into the enterocyte by the apical membrane transcellular protein divalent metal transport 1, or DMT1. Iron within the cell can also be stored in its storage form ferritin if it is not needed, and is removed as enterocytes have a lifespan of about 3 days. When iron is needed, it is exported by ferroportin, which is found on the basolateral membrane. When the body is replete of iron, iron absorption and exportation is blocked by antimicrobial peptide hormone hepcidin, which is produced by the liver. Hepcidin binds to ferroportin and forms a complex, which is degraded internally, preventing iron in the enterocyte from leaving. Hepcidin may also decrease DMT1 expression. But let's go back.
In order to bind to transferrin and go through circulation, ferrous iron is oxidized back to ferric iron by the peroxidase enzyme hephaestin. When ferric iron is bound to transferrin, a protein found in serum, it is kept in a non-reactive state when bound. When transferrin is fully loaded with 2 atoms of ferric iron, it is called diferric transferrin. Diferric transferrin is transported through circulation and binds to transferrin receptors, which are found on the cell membranes of erythroid tissue, forming a transferrin-transferrin receptor complex. This complex is internalized in a vesicle coated with clathrin, which unloads and is recycled back to the erythroid tissue cell membrane. DMT1 and proton pumps are also found within the vesicle membrane. The proton pumps then acidify the inside of the endosome by pumping hydrogen ions inside. Because of the decreased acidic pH, the ferric iron is released from the transferrin-transferrin receptor complex. Because DMT1 exports ferrous iron, ferric iron needs to be reduced. Within erythroid tissue, ferroreductase enzyme STEAP3 reduces ferric iron to ferrous iron, which is then used by the tissue. The unloaded transferrin protein is now called apotransferrin, and, along with the transferrin receptor it is complexed with in DMT1, is recycled back to the erythroid cell membrane. At neutral pH, apotransferrin dissociates from transferrin receptor due to low affinity and re-enters circulation to reload iron.